Bismillahirrahmanirrahim. I'm Dr. Faryl, working as a assistant professor in the University of Veterinary and Animal Sciences, Lahore. And first of all, I'm very thankful to the organizer for giving me this opportunity so that I can come over here and I can share my research expertise with on this esteemed forum. Today, I'm going to talk on one of the important research topic, that's the molecular characterization of the avian species of Punjab province using uh, mitochondrial genes. And like first of all, the thing is that the introduction regarding the avian biodiversity, that is a very important one. And as you people know that the birds are regular invertebrates that belong to the class Aves and that have the feathers, wings, and the move by means of the two limbs, and they are highly significant and diverse component of the ecosystem, as they have an important role in the maintenance of the ecosystems, by playing important role in the seed disseminations by acting as predators, prey, and by pollinations of the plants. Many birds are rarely of the economic importance as they are a source of the food and are used broad range of the Pakistan is remarkable ecological regions of the world. And Pakistan is actually harboring a diversified area of the fauna due to the variety of the habitats. And among the diversified avian fauna, the most striking features are the pigeons and the parrots. They are very important uh, bird species. And they let's talk about first of all few words about the pigeons. They are monumentally important and oldest domesticated bird, and they have played a very significant role throughout the human history, including the symbols of the god, pets, and the food companion animals, messengers as well. And pigeons trading is a lucrative business worldwide, and there is a lot of breeds of the pigeons, like including the Bangi, the lucky pigeon, the Shirazi, the rock pigeon, the dengue, the Chatri, Lotan, etc. And they actually one of the problem, just like that of the pigeons, the parrots are among the most striking features of the tropical avian fauna, and their ability to talk and intimate human voice makes them love birds and increase their popularity as a pest. And as far as the species of the parrots are concerned, Pakistan have more than worldwide there are more than 370 species of the parrots. And but the main problem with the identifications of the conventional ways of these two breeds and species are that most of the time we people mostly use the traditional approaches for their identifications, like we identify them, we classify them on the basis of the body weight, the skin color, the eye color, the comb type, the marks, and the necks. But all these approaches are the good because they are also going to be affected by the environmental factors. So molecular characterization approaches are comparatively more authentic, including the randomly amplified polymorphic DNA analysis, restriction fragment length polymorphic analysis, microsatellites analysis, and mitochondrial genome. And among them, mitochondrial genome having a very significant positions over there. And as first of all, now a little bit about the avian mitochondrial DNA genes. And as far as inherited pattern is concerned, that is exclusively maternally inherited, and it's going to play a very important role in the evolutionary study. In the phylogenetic analysis, mitochondrial genome play a very important role, and because there is no recombinations in the genes, and they are in the present study, the pigeons breeds and the parrot species, they have been classified by using the mitochondrial genes. First of all, the two birds were selected through breeding, conventional identification methods, and then the DNA blood was collected by to the process of the phlebotomy by using their with small insulin syringe, and the DNA was extracted from that sampled blood by using the organic methods. And then for the amplifications of the two regions of the mitochondria, cytochrome B gene and ND2, the primers were designed by using the online software followed by the PCR amplifications and sequencing. After the sequencing by using the bioinformatic softwares, bioinformatic analysis was done. This map actually depicting the different regions of the Punjab from which the extensive sampling has been done for these studied pigeons and breeds and the parrot species. After the sequencing, the final haplotype was constructed, and you can see over here that the final haplotype will also and sequence have also been submitted and deposited on the NCBI repository. And you can see over here the haplotypes of the pigeons that there are five main uh, mm, points or polymorphic sites, which is going to be differ, which actually differ from the already published and sequenced data. And same, the ND2 genes haplotype of the pigeon is also depicting the fewer polymorphic sites. And actually, these polymorphic sites are going to be are rendered as the molecular marker for the identifications of the pigeons from the rest of pigeons of the local pigeon breeds from the rest of the world's breeds. And the same on the other hand, the cytochrome based B gene based sequence B gene based haplotype was constructed for the parrot species, and the red highlighted are the polymorphic sites in the cytochrome B regions of the parrots. And parrots activity show that this is a highly polymorphic site, and the region in which there is a numerous polymorphic sites. Actually, on the basis of the haplotype, the phylogenetic analysis shows that Columbar pestris is the deep common ancestors of the Pakistani rock and Pakistani domestic pigeon breeds. And through green route from the Siberia, they enter into Pakistan through Afghan west border and in India through the Ganga flyways. And from Bolivia, the immediate ancestors of the Pakistan species. And as far as the parrots are concerned, Pakistani rosy parakeets are uh, contagious taxa of the Indian Citicola comari melinensis and the district from the Indo-Pak Citicola comari borealis. And Citicola Euryptera meningorostris is a deep ancestor of the Pakistani Alexandrines parakeet. That is vernacular of this Alexandrine parakeets is actually the raw parrot. And thank you very much for your complete attention. Actually, this is the these studies and these findings can be helpful for the for the breeders of these two species, and they can solve the problems. They can overcome the problems of the conventional methods of their identifications, and as far as they can also help out the breeders for their further identification, so that the best species and the breeds can be made for the next progenies. Okay, thank you very much.